So this video is going to go into a little more depth on touch perception specifically, and then it'll touch a little, no pun intended, on active touch, also known as haptics. First question you might ask about a sense like touch is, what's its absolute threshold? Like, how sensitive is the sense of touch? So for humans, if we're using the pads of our fingers, we're actually able to detect a little dot that is 1 micrometer high on an otherwise smooth surface. That is 1/25,000th of an inch. Now, human hair is 100 micrometers wide. A single red blood cell is about 8 micrometers wide. And we can just barely feel 1 micrometer on a smooth surface. So remember that next time you're struggling to find where the line on the toilet paper or the packing tape is. As I've said, though, that's when we're using our fingers where we've got great sensitivity. We actually have lower sensitivity on other parts of our body, like our back and our stomach, which, as we saw before, also have proportionally fewer neurons up in the somatosensory cortex representing them. However, we can't actually distinguish two very fine textures. Like, let's say a texture that has 10 micrometer elements versus 100 micrometer elements. That's certainly larger than our absolute threshold. We should be able to detect it, but we actually can't unless we're moving our finger across the surface. We actually need the vibration information to detect something so fine-grained. Indeed, experiments have shown that if we temporarily shut down Pacinian corpuscles and only Pacinian corpuscles, that actually shuts down our ability to feel those fine texture differences. So normally we can tell the difference between those two textures by rubbing our finger across it, moving our finger across the surface. We can tell whether that really, really fine-grained sandpaper is different than that other really, really fine-grained sandpaper. But without Pacinian corpuscles, suddenly we can't do that task. But now let's take a moment and think about some ways we might measure how touch perception works. Like, how do we actually do the measuring to figure this stuff out? Well, for static touch, you'll remember that Merkle discs are our heavy hitter. They're the really important mechanoreceptor for static touch. And we know there are a ton of densely packed Merkle discs on, say, the fingertips. So we have really great tactile acuity there, like touch acuity there. So what you can see here are a couple of common ways we use to measure acuity on different parts of your body. So you could do the fingertip, but also, as you see in the picture here, you could do the back of your finger, the dorsal side of your finger, kind of the top there. Or you could do halfway up your arm, or you could do your back, or whatever, and compare different parts. Of course, it'll be different at different parts of your body. Here on the left, the first picture labeled A, is how you find what's called a 2-point touch threshold. So basically you poke the skin with either a single point or simultaneously with 2 points that are a certain distance apart. And you can change that distance, right? And the recipient isn't looking. So they have to go off of touch alone and tell you, the participant tells you whether they felt 1 poke or 2. Right? And they don't know if you're doing one of those fake trials where you just poke them with a single poke, or whether it's 2 different pokes at a certain distance apart. They just have to say whether they felt a single touch or 2 separate touches. And on the trials with 2 points, when they're like really far apart, it's easy to tell the difference. It's easy to tell that it's 2 different touches. But when they're close enough together, people feel it as a single point and actually get it wrong a lot. Like they're being touched by 2 separate things in 2 separate places, but they think they're just being touched in a single one, and they say it's a single touch. So that threshold, the 2-point touch threshold, would be the smallest gap between those 2 points that a person could reliably detect it as 2 separate touches, like 50% of the time or more. So about as close together as you can get where people are still able generally to say, oh yeah, that's 2 separate pokes. Picture B, to the right of that, is just a different way of measuring that same basic idea. In this case, you press like a metal grating against the skin, like you see in the picture here, where you can line that grating up either with the grooves going one way or going the other way when you press it against the skin. And the recipient just has to tell you how it feels, which direction they think the grooves are pointing. Now, if you do this with big old wide grooves, it's pretty easy. But with really small grooves, people suddenly can't do it. They guess wrong, or they're not sure which direction the grooves are going. And we find that the smallest grooves that someone can reliably get right about 50% of the time, and that is the threshold. That's their tactile acuity. And it turns out the tactile acuity, meaning the acuity for touch, is different on different parts of our body, as you can see here. Like, here's an example, some real data of that difference for just your hand. Of course, we could do it for the whole body. So note the y-axis on the side here. The y-axis, that is tactile acuity measured in millimeters, where smaller means better acuity, right? Fewer millimeters means smaller distances where you can still accurately detect that it's two separate points touching you. The fingertip has the best acuity, as you can see here, less than 2 millimeters on average for humans. Well, it's a little worse at the base of the finger and quite a bit worse on the palm. Other areas of the body are even more extreme, where 2 points can be pretty dang far apart and it still feels like a single touch. But now note the x-axis at the bottom there, at the bottom of the graph. It's showing the average spacing between one type of touch receptor at that part of the body, specifically what they label here the SA1. That means slow adapting 1, meaning the first type of slow adapting mechanoreceptor, which is the first type of slow adapting mechanoreceptor we learned about, which would be Merkel discs. So they didn't label it here as Merkel, but that's what they're talking about. They're talking about how far apart the Merkel discs are on the fingertip versus somewhere else. And on the fingertip, those tend to be really close together. The fingertips are dense with touch receptors, but other parts of our body, not as much, right? They're further apart. You don't have quite as many Merkel discs all close together. Now, you'll remember we ran into the term receptive field in a previous video about the auditory sense, where I mentioned the RF, the receptive field of a neuron, is just whatever proximal stimulus affects its firing. In other words, whatever it is that turns this neuron on, whatever it is this neuron is tuned to. So for a particular Merkel disc, it goes off only for touches, for pressure in that tiny area of skin that the Merkel disc is embedded in. So the receptive field of a Merkel disc is that little area of skin right above it. Right? You can kind of see in the graph here, this might be, here's one Merkel disc, one neuron, and you can see it's kind of watching this one patch of skin here. Here's another one, here's another one, here's another one. But they've dotted the outline here saying this is the patch of skin that this particular neuron is watching. And yes, it might overlap, right? So if someone touches in this part of the skin where my mouse is right now, both of those two neurons might fire a little bit, right? So that the brain could tell it's somewhere in that region. But again, the area watched by one Merkel disc, this is like the only area of the skin that if you press on it would make this neuron go off. Indeed, if we go back to those 4 mechanoreceptors we learned about for touch, we already know some basic facts about them. So first off, we remember the ones that start with M, Merkel discs and Meissner corpuscles, those are near the surface of the skin. Whereas the ones kind of with letters deeper into the alphabet, Pacinian corpuscles or Ruffini corpuscles, those are deeper in the skin. And we learned that some of them are rapid adapting, right? Meaning that they only fire to changes in stimulus or the start and stop of a stimulus, whereas others are slow adapting, meaning they keep firing as long as the stimulus is there, right? So that was our Merkel discs and our Ruffini corpuscles. But now we can add one more fact that kind of makes sense when you think about how deep in the skin or how near the surface these different types of receptors are. Specifically, how big the receptive field is for each type of neuron. How big of a patch of skin that neuron happens to be kind of watching and affected by. We talked about Merkel discs, and you can see an example from someone's fingertip here, right? And a Meissner corpuscle is pretty similar, maybe just a bit larger. But what about the receptive field of those deeper mechanoreceptors, deeper down in the skin, like a Pacinian corpuscle? Well, their receptive fields are going to be much larger and wider and more diffuse. And think about it, it kind of makes sense. Detecting vibrations that have propagate it all the way deep into the skin means it won't be nearly as precise about exactly what bit of skin nearby received the vibration, right? You can vibrate a little ways away from where the actual Pacinian corpuscle is, but the vibration's traveling down, deep down into the skin. So even if it was off to the side a little bit, it might reach the Pacinian corpuscle and turn it on, not as strongly, right? So it's kind of weaker, it's more diffuse along the edges there, but we'd say it has a larger receptive field. Right? It can pick up vibration from a larger area. And similarly for the Ruffini endings, which again are also deep in the skin. So they're detecting skin stretch, but affected by skin stretching kind of anywhere nearby in the surface, not just right exactly above it. Here's just a different depiction of the exact same thing. Again, we can pick out the types of mechanoreceptors that are near the surface. So our Merkel discs and our Meissner corpuscles here. And you can see with the ones near the surface, like the Merkel discs, you can see an individual neuron's receptive field is very tiny. It's basically just a little point on the skin where it'll detect pressure there. So there's one neuron here at the tip of this finger that we're talking about, and here's its receptive field, just this little purple dot, okay? But then compare that to the large, vague, fuzzy receptive fields of a Pacinian corpuscle or a Ruffini corpuscle, where they've drawn a big blue area shading surrounding, showing all the places where some vibration could turn on this one neuron here. Or likewise, some skin stretch anywhere in this blue region on the index finger could turn on that one Ruffini ending there in the middle of the index finger. So larger, more diffuse receptive fields, not as precise. If you want to try this stuff out at home, kind of test your own acuity, it's pretty easy to test your own 2-point touch threshold if you have a friend or a partner who can help you. If you want decent precision, you can even get a sewing gauge like this. It just costs a couple dollars, or like a sliding ruler kind of device so you could tape a couple paper clips sticking out of. But if you're just doing it for fun and don't worry too much about the measurements being precise, you could even just bend a few paper clips so that the two tips are different distances apart on each paper clip. So get some where they're really close together, some where they're a little further, some where they're a little further, and so on. And maybe keep one paper clip out where it's just a single point. So you can do control trials where there aren't actually 2 points, where you'll just be touching one single point. And then the person being tested, you could take turns with this, right? But the person being tested closes their eyes, and the friend or partner, the person being the experimenter, will test the different paper clips in random order at some place on the body. Like maybe to start, you could do the pad of your fingertip, or after that you could try the underside of your forearm, or on your calf, or on your cheek, or whatever. But test out different parts of your body, and you'll quickly find vastly different performance on that 2-point touch threshold in terms of where people start getting it wrong and being confused about, oh, that felt like a single touch, but actually, the paper clips might have been an inch or more apart when you're testing your calf or your back or something like that. Here's some actual data averaged across a bunch of humans where we do this 2-point touch threshold. Note that the y-axis on the left here, it's in millimeters. It's how many millimeters the 2-point touch threshold is. So the smallest gap you can reliably tell is actually 2 distinct points, meaning the smaller threshold is better. Smaller threshold means better spatial resolution. Notice then that the best resolution is in the fingers, right? We got all our fingers here, and on the face, like by our lips and stuff like that. So compare that to how well people do on their calf or their thigh or their upper arm. 50 millimeters means 5 centimeters. It's about 2 inches. You can touch someone with 2 paperclip points that are 2 friggin' inches apart, and half the time they can't even tell it's 2 distinct pokes, and they'll say they just felt a single touch. They think it was a single paperclip. Now, the really important and interesting thing to note here is that this all strongly correlates to that homunculus we talked about in S1, in the primary somatosensory cortex. Places that we have better spatial resolution usually means we've got more sensory receptors in that part of the skin, but also more neurons in the brain that are dedicated to processing that area. Now, at any given place in our skin, Merkel discs usually give us pretty good tactile acuity relative to something like Pacinian corpuscles, because those Merkel discs have small and precise receptive fields, not those big fuzzy and vague RFs that are found for Pacinian corpuscles. You can see how small RFs are helpful if you look at this picture, right? The green arrows, that's like our 2 paper clips a certain distance apart in both of these cases. Now, if each neuron has a kind of a small personalized receptive field, it only watches this small area of skin, and then if it's poked, it sends a signal off to the brain, sends a little signal down to the brain, says, I was poked, right? So if they have these small receptive fields, like on the right, then it's easy to see how the brain could tell when there are 2 touches. In this case, both neurons would go off separately, and the brain goes, oh yeah, there's 2 pokes, I feel 2 touches. But with a big receptive field, it's hard for the brain to know if 1 touch or 2 touches are in that big red area, because it goes off the same either way, right? This neuron fires to the brain, and all the brain knows is something's in this area. This neuron fired, so something's out there in this area, but it doesn't know whether there's 1 or 2 things there, right? It can't as easily tell the difference between them. But also, in some areas of the body, we have lower acuity, lower spatial resolution, worse performance, because a bunch of receptors are all hooked up to a single brainstem neuron heading toward S1. This is actually called convergence in the neuroscience literature. This convergence, where a bunch of neurons, these would be like a Merkle disc watching here, a Merkle disc watching here, Merkle disc watching here, and so on, but all of them just go to this single neuron in the brainstem that'll then head on to the brain. This convergence means that when that brainstem neuron fires, this one down here, when it goes off and the brain gets a signal from it, the brain doesn't know which of the hookups were actually activated to make it fire. It doesn't know if you were touched in this green area or this area or this or 2 or 1 or any of that. Basically, we would say that brainstem neuron, the one down here, has a larger and less precise receptive field than each of those individual neurons that's attached to it. So here, imagine like this one highlighted in green here, that's a single Merkle disc watching a little patch of skin. So that single Merkle disc has a nice small receptive field. But this brainstem neuron also has a receptive field. In this case, the patch of skin that this brainstem neuron is kind of watching or affected by, if touch happens anywhere in this big combined area, that would make this neuron go off. So you'd say it has this big, large receptive field combining all of those smaller components that come from all these neurons that synapse onto it, that converge onto it. Whereas in other parts of the body, like on our fingertips, right, where we need more precision, we actually have much better acuity. And one reason for that, partly again because we have more brain neurons that'll process that area as well, and just more receptors there. But also an additional reason is we have less convergence. Each individual Merkle disc might get its own private brainstem neuron heading up to S1. So here's a Merkle disc watching this little patch of skin highlighted in green here. But notice it's the only neuron that synapses onto this brainstem neuron that again then goes on to S1, the somatosensory cortex, which means if this neuron goes off, if the somatosensory cortex gets a signal from it, then we know exactly where on the skin. It must have been this tiny little patch here that got touched. So we can be very precise about exactly where the touch happened if there's not as much convergence. So there's less convergence in some of the wiring for some parts of our body, like the fingertips or the lips. That signal to the brain is going to be more specific about exactly where on the skin a touch happened. It means that the brainstem neuron gives more specific info to the brain then. So we would actually say each of these brainstem neurons then, at the bottom here, has a smaller and thus a more precise receptive field. So the receptive field of this brainstem neuron, when you think about what makes this green one on the left fire, the only thing that makes it fire is touches to this little patch of skin here. So its receptive field is that little patch of skin. That's how much. That's the area of the body where stimulation can affect this brainstem neuron firing. So again, it also has a receptive field. It's not just our sensory neurons, like the actual Merkle discs, the mechanoreceptors that have a receptive field. All of the neurons in our sensory pathways have a receptive field. You just have to think back to what makes this neuron go off. Well, it's only watching that little patch of skin. What makes this other neuron with all that convergence go off? Well, it's this huge patch of skin. Anywhere in there can affect the firing of that neuron. Here is just another diagram to try and make that idea of convergence more clear. So here we might have 3 neurons. Maybe these ones are thermoreceptors or whatever, but each one has a receptive field of some patch of skin. The right type of stimuli happening on its patch of skin makes that neuron fire. So one of the neurons, right, we can see it here kind of highlighted in blue. One of the neurons watches the blue patch of skin, one watches the yellow patch of skin, and one watches the purple patch of skin. And yes, they might overlap a little bit, but notice here the wiring is convergent. So the 3 neurons all synapse onto that one later neuron, which then sends signals up to S1. That means when S1, the primary somatosensory cortex up there in the top of the brain, when S1 gets a signal from this single wire, it has no idea whether the stimulus happened in the blue or the yellow or the purple zone, just that it's somewhere in the combined region. So if I ask you what's the receptive field for this neuron, this second neuron, right, the one that's passing the signal along, the receptive field for it would be the combined region of skin, what they've kind of highlighted all around this big area here in the solid blue line. That would be the receptive field for that neuron, that later neuron. It's much less precise. Here's my own drawing of the idea, and then we'll move on. If you can forgive my drawing skills. First off, I've drawn 4 Merkle discs off to the left here, Merkle disc 1, Merkle disc 2, 3, and 4, and outlined their receptive fields with just some black circles, like that's the patch of skin they're watching. Again, in reality, they might overlap a little bit on the skin. And then you can see each one sends its axon to a neuron up in the brainstem, which I've labeled A, B, C, or D, right? And what's the receptive field of each of those neurons? Well, each of the brainstem neurons has the same receptive field as the Merkle disc feeding into it. So you can see I've drawn a little dotted circle around Merkle disc 2's receptive field and labeled that's B's receptive field. Again, horrible handwriting, but that's B's receptive field there, okay? That means the RF of brainstem neuron D would be the same as the RF of Merkle disc 4 in this particular image. And then each of those brainstem neurons goes up to S1 in the brain where they arrive. Again, they'll pass through the thalamus on the way, right? But then eventually they reach S1 up there where they arrive at one specific neuron, E through H, or whatever I want to call them, right? In this case, when does H go off? Well, H will only go off if D went off, which means H only goes off if Merkle disc 4 went off, right? Because that's the direct pathway here. That's the only thing that turns on H. So when H goes off, or when E goes off, the brain knows exactly what bit of skin was touched. It's got good precision if we have this nice kind of private wiring where there is not convergence, right? There's no convergence here. But compare that to this other drawing I've added at the bottom where I put in a lot of convergence, where we do have convergent wiring. Now each Merkle disc here still has the same size of receptive field, the little bit of skin above it that it's watching, but they all synapse onto A in the brainstem, this one neuron. And then A's receptive field would be the whole combined set of areas. So I drew a blue dots all around the whole combo here. Blue dot, blue dot, blue dot, all the way around to say A's receptive field is that whole combined region, right? And that means when A passes its signal onto the brain and hits a neuron in S1 in the brain, like neuron E here, then E also has that big combined receptive field. E will go off if there's a touch anywhere in that combined big area. So I drew green dots all around here for green neuron E over here to show its receptive field. It has a big, large receptive field because it's that whole combined area. That means if some touch stimulus occurs anywhere in that big combined region, like any of the places with red X's that I've drawn here, whether it's one stimulation or two, like 2 red X's at the same time, then E will go off. But that means when E goes off, the brain, in other words, has no idea where in that combined region the touch was. It can't tell apart the top X from any of the bottom red X's. It can't tell whether there was a top X and a bottom X or just a top X, right? In all cases, it's just turning on A, which turns on E. So that makes it less precise. It has less acuity. Now that we're kind of getting more of a feel for using that term receptive field, we can see that what we were describing earlier about the primary somatosensory cortex in previous videos was actually just the receptive field of a bunch of neurons up there in our brain, up in S1, right? What Wilder Penfield discovered when he was mapping out the somatosensory homunculus was basically the receptive field of those neurons in S1. In other words, which part of the body, which patch of skin each neuron up there in the brain was specific to, which bit of skin, or maybe even a large patch of skin, it normally gets its signals from. Now, some neurons in S1 have small receptive fields. Again, perhaps fed directly by a single Merkle disk's signal if there's no convergence, if it has its own private wire. But others may have larger and kind of overlapping receptive fields, and thus it might fire for a touch to kind of anywhere in that bigger region. The brain then perceives it with less specificity too, meaning we'll get less acuity when we measure what you feel. When I ask you, did you feel 1 or 2, like with the 2-point touch threshold methodology. So you can see an example here for a monkey model. For monkeys, we do a lot of the neuroscience research earlier with animals before humans. But the outlines in A, B, and C here are receptive fields for specific neurons in the monkey's brain. So what you're seeing, they're not showing you where an individual Merkle disk is that has this giant receptive field. Rather, they're saying there's a little neuron up in the monkey's S1, up in its somatosensory cortex, and we can say one of the neurons up in its brain watches this little patch of skin on a finger, this little patch of skin on a finger. But there's another neuron up in its brain that might watch a much larger area on the back of its hand, and maybe even a giant area on the back of its arm. So some S1 neurons are very precise, small receptive fields, like if they got a signal via a single direct wire from a single Merkle disk on the finger, whereas others are more big and vague because they got a signal coming from convergent wiring, maybe coming out of a bunch of Merkle disks in the forearm, and probably there are fewer Merkle disks overall in the forearm. And then we've got like way more neurons up in the brain representing the fingers than the forearms in order to keep track of those minute differences and that density of receptors out in the skin. It turns out the initial neurons in S1 that get signals from the thalamus, right? So just the S1 neurons, the main S1 neurons, they tend overall to have pretty small receptive fields. But when we look at the receptive fields of neurons further away from S1, in other parts of the brain where we start to process touch signals and bodily signals in more bigger picture ways and things like that, we start to find neurons with very broad receptive fields. For example, we find a neuron that might fire for touch to any of the fingertips, right? A neuron that goes off specifically when you're touched here or here or here or here. In other words, if any of those Merkle disks across all those fingers go off, then there's this one neuron later in your brain, again, maybe in like S2 or some other area, that'll go off to just say, I'm touched on a fingertip somewhere, not a specific fingertip. Or one that fires for a touch on any finger all the way down for a specific hand, maybe just for your right hand or just for your left hand. Or a different neuron later in the process, later in the cortex there, if we recorded a different site in your brain, a different fold of the brain there, we might find a neuron that goes off for a touch to the fingers of either hand, right? So it doesn't matter if it's left or right. Basically, you can think of that as hundreds and hundreds and hundreds of Merkle disks all across those different areas. But if any of them fire, then this one later neuron watching for that will go off and say, hey, somewhere on my fingers I was touched. It's kind of specific to finger touches. So in other words, later processing in the brain will generally get more big picture, more general, building up from small pieces of info early in the process. Maybe a neuron only fires for some combination of inputs later, or maybe it fires these later neurons if anything in the combined region happens, like with the hand RFs here. Kind of like how in A1, right, the primary auditory cortex, it started with neurons that have a precise frequency of sound they fire for, a very simple input for each of those neurons, a simple receptive field. But other areas further into processing, further out into the brain, like in A2, the secondary auditory cortex, right? It fired for combos, more complex combinations or types of combinations of frequencies, like when we learned about pitch neurons. Same kind of idea here, and same thing we'll see in other sensory systems. The brain starts with picking up simple pieces to have receptive fields firing for very simple stimuli, and then we have later neurons further into the brain that fire for more complicated combinations of those things. Now, all this wiring, all this layout is pretty damn impressive, but it's important to understand that it also builds and changes with experience. What we call neuroplasticity, which is just changes in neural wiring, like where synapses form and stuff like that, based on our experience. For example, here is a classic study using brain scanning to look at how various body parts were represented in S1 for people who had played string instruments for a long time with their left hand. So think about it, like someone with a violin, that means they're doing the fine-grained precision finger movements with their left hand, while their right hand only has to do more gross motor movement of the bow that they're holding. On the right hand, it doesn't get as much practice or experience using the individual fingers of the right hand compared to controlling all those individual fingers very distinctly on the left hand. Okay, so what did this study find? Let's just read the abstract here. Magnetic source imaging, so brain scanning, revealed that the cortical representation, right, so in the cortex, they mean S1 here. So the representation in S1 of the digits of the left hand of string players was larger than that in controls. In other words, people who didn't play string instruments for a long time. The effect was smallest for the left thumb. That makes sense. Like a violin player isn't going to use their thumb as much. And no such differences were observed for the representations of the right hand digits, right? Because that's just doing the gross motor movement of the bow. You're not using your individual fingers all that much on the right hand. And, more impressively, the amount of cortical reorganization, in other words, the extent to which the brain had reorganized things and devoted more neurons and more connections to each of those individual left fingers, the amount of that reorganization in the representations of those fingering digits was correlated with the age at which the person had begun to play. In other words, the number of years they had spent practicing. The more practice, the more the brain had reorganized to give more neural representation, more processing power to each of those fingers. So these results, they say, suggest that the representation of different parts of the body in the primary somatosensory cortex, that's S1, of humans depends on use and changes to conform to the current needs and experiences of the individual. So the more you use those fingers and get feedback from touch and feedback from other modalities like hearing the sound it makes, the more your brain dedicates neural real estate to that task. Likewise, acuity of a fingertip actually improves if you use it to do fine-grained discriminations a lot, like with braille reading. And acuity of a fingertip gets worse if you have it in a splint for a few weeks and you don't use it to touch things. It's all due to adjustment and reorganization of connections up there in the cerebral cortex, in S1, in the somatotopic map of our body that we have up there in the brain. In other words, it's due to neuroplasticity. Now, I wanted to take a break here from all this brain stuff and mention a few fun demos you can do at home to sort of memorably experience some of these things. So the first one, it's fun but requires a friend, someone you're okay letting touch your forearm. What you do is the subject, you can take turns doing this, but whoever's going to be the subject closes their eyes and lays their non-dominant arm extended straight out with the palm face up. Just like rest the back of your forearm on a table comfortably, kind of like you see here, okay? Then close your eyes and keep them closed for this whole trial, this whole experiment. The friend, acting as experimenter, will then very slowly trace their finger with the lightest of touch up the subject's arm, super slowly, going from the palm, like the wrist area, so right about here, just going up really slowly, really lightly, very slowly, until they get to the crease of the inner elbow. So in other words, the end of the forearm. Don't start going all the way up the rest of the upper arm. Use a light touch here, a steady pace, and don't rush it. Now, the subject, the person being experimented on, the person with their eyes closed, you just have to say now when you think the finger has reached the crease of your inner elbow. When you feel the finger get to that crease of your inner elbow, say now as soon as you feel it there. What you'll find if you try this yourself or on a few friends is that most people are not accurate at all. Acuity is really poor in our arms relative to what you might think or expect. You don't realize how imprecise your sense of touch is on many parts of your body when you get away from things like your fingers or your lips. Okay, another similar demo that can be kind of fun to try at home might only work if you've got a partner or a friend that you're comfortable with, because this one involves bare feet. In this case, the subject, the person being experimented on, the subject takes off their shoes and socks and sits with their bare feet solidly on the ground. Just sit there comfortably in a chair or the edge of the couch or whatever. have your eyes closed or be blindfolded throughout. So just keep your eyes closed for the whole thing. And then the partner or the friend, the person who's acting as experimenter, then is going to take turns very lightly and briefly touching one of the middle 3 toes with the tip of a pen. You don't have to use your finger, just use the tip of a pen, something really small, and just lightly and briefly touch one of the middle 3 toes. The person being tested then has to say aloud each trial what toe they think was just touched. It may help to number them first, like 1 means the big toe, 2 means the next one in, and all the way to 5 toes. But again, for this, just do it for the middle 3 toes, because that's where people have the most confusion. The subject keeps their eyes closed, and you'll just repeat this for a bunch of trials without giving any feedback along the way of how they're doing. And what happens when people do this, if you try this out, generally mistakes are very common, much more common than you would expect. The receptive fields of individual receptors on each of those toes may be pretty small, right? The Merkle discs down there. But the wiring coming out of there actually converges together a lot. Many of those Merkle discs converge together onto a single neuron, passing the signal onward into the brain. So what the brain gets is a much more fuzzy and vague signal to guess from. You can feel the convergence in action by how poorly you do on this task. But with additional practice, and especially with feedback, or even just doing it while watching where you touch, your brain will get a little better at distinguishing any small differences in the signals, like in the ratios of which neurons with which receptive fields were slightly affected or largely affected in their firing rate. And you can become a little better at this due to that neuroplasticity, do that practice. All right. These next 2 demos I'll talk about that you can do at home, they don't technically fall under touch, but rather under thermoreception. This seems like a good time to mention a couple of the demos for sensing temperature changes and specifically help us see the receptive field of your thermoreceptors now that we've talked about receptive fields a lot. So I call this first one cool spot density because it's a quick and easy way to map out the density of those thermoreceptors that are sensitive to cooling. In other words, to when it gets colder on some part of your skin. Sometimes we call them, or the little bit of skin that makes up their receptive field, a cool spot. So you'll find where your cool spots are for a couple little patches of skin on your body. First off, for this one, you'll need a friend or a partner or someone to help you out to assist with this one. And it helps to ahead of time grab some paper and a ballpoint pen or 2. And on the paper, what you want to do to start is just draw a finger, kind of like you can see here. Just draw a finger with a little, 5x4 grid on it, like you see here. Just 20 little boxes. The total area, if you think about this being on the back of someone's finger between those 2 knuckles, we're talking about roughly a square centimeter, right? A little different for everyone, but roughly a square centimeter. But just imagine that grid on the back of your index finger between those 2 knuckles. Now your friend, once you're ready, you'll set your finger down, maybe just lay it gently on a table or something so that the back side of the finger is up like this. Your friend will use the tip of a ballpoint pen to lightly touch the skin on the back of your finger at each of those 20 points within the grid. You don't actually have to draw the grid on their finger. Just touch roughly here, and then here, and then here, and then here. Like do 5 touches, and then go a little down and do 5 touches in a row, and so on. But for each stimulus presentation, this will take a few minutes, because for each time that they touch the pen down, they should slightly depress the skin and keep the probe, keep the tip of the pen in place for about 2 to 3 seconds. Then they remove it. And after removing the pen, the participant just says yes or no for whether they felt a cool sensation. So if that tip of the pen felt kind of cool, felt kind of coldish, then say yes. If it felt neutral, if it didn't feel cool, if you didn't have that experience, that sensation, then say no. And ballpoint pens actually work well for this because they're a good heat sink and they're cooler than your skin. Plus the tip is small enough, you'll likely only stimulate one receptive field, one cool spot's receptive field at a time. It helps to actually leave the pen off your skin for a good 5 seconds or so between each trial to ensure that it doesn't get up to skin temperature. And then on each trial, your friend should just mark on the piece of paper, on the data sheet. They can just mark a little X on the paper grid or fill it in or put a dot anywhere that they discovered a cool spot, anywhere that you said yes. And at the end of this, you can count up how many there were in that patch of skin on the back of your finger. Then after that, what you might do is draw another grid of the same size on the paper, and this time imagine it projected onto something like the underside of your forearm, about halfway up, like halfway between the wrist and the crease of the elbow, like halfway up on the underside of your forearm. You could do that same thing. Have someone touch, just gently, lightly, for 2 to 3 seconds, touch a pen to each of those little spots on that imaginary grid and ask you, does it feel cool? Did you feel a cool sensation? Sometimes you will, sometimes you won't. You just say yes or no, and they'll write down which ones. And again, you can record how many cool spots you find in that region of your skin. And you can try it elsewhere too if you want. And of course, you might want to switch so the other person can experience it as well and map theirs out. This is actually, when I've done this with a small group of people before, I wrote the numbers down. They came out looking like this. So averaging about 9 cool spots on the finger, the back half of the finger, and a little under 8 on the under part of the forearm. So slightly different density. And if you try other parts of your body, you'll find even bigger differences. Okay. And again, this is a small sample size, not perfectly representative. You might find slightly different numbers for yourself or your friend. Of course, what we really want, if we want to know humans overall, would be take a large sample. And someone has done this. In fact, a long, long time ago, people did this. What you saw a minute ago in my little sample is actually pretty close to what we would expect from the classic published data, which actually goes back to the 1920s and 30s, and those same results hold up now. But they've measured both cool spots or cooling receptors and also warm spots or warming receptors. Because remember, we have thermoreceptors for both, for temperature going up or temperature going down on our skin. And classic measures in humans, the data here, find about 7 to 9 cool spots on the back of the finger. Remember, I found about 9 in my little sample. You might find something similar on your own finger, but right finger dorsal means like the back, the top, if you're holding it like that, like a dorsal fin would stick out the back there, right? So the top of the finger where we did it, whereas the forearm, right, we did the underside of the forearm, so it might be a little different, but the forearm in various places has about 6 to 7.5, right? And I found a little over 7. So compare that to something like the sole of your foot, where you only have a few per square centimeter, but on your lips, you have like 16 to 19 cool spots in a single square centimeter. And almost, in other words, in that little grid of 20 little spots, almost all of them would have its own separate cool spot, its own little thermoreceptor. So on your lips, much more precise for detecting coolness. Now notice we've got way fewer warm receptors overall, much less density of warm receptors. In fact, if you look at the drawing over here on the left, this is the drawing of someone's forearm over here, and the bigger open triangles, so you can see a bigger, like an open one here. It's kind of hard to see in this drawing. It might not even look like triangles. Maybe it just looks like big open circles here. But those are where they found warm spots or warming receptors, warming thermoreceptors. Whereas all the little dots all over the skin here, those are the cool spots, the cooling receptors. Okay, one last demo. There's another thermoreceptor demo, but one last demo you can try at home before we move on. This one's really, really simple because all you need for this is 3 pennies. So what you'll do to prepare for this is put 2 of those pennies in your freezer for a while, like 10 minutes at least. You want the metal to get cold, okay? Leave the other penny sitting out at room temperature. And when you're about ready to do the demo, you'll actually want to take that third penny, the room temperature penny, put it in your hand for a minute, like hold it in your hand, maybe even between a couple of fingers, because you want it to get near body temperature. Okay, so what you're going to have is 2 cold pennies and 1 that's body temperature. Then for the demo, take those 2 freezer pennies out and lay all 3 pennies in a row, touching like in this picture here, on a flat surface like a counter or a desk or a table. Put the cold ones on the outside and the body temperature one in the middle. And then with your right hand or whatever your dominant hand is, take the middle 3 fingers on the hand, your middle 3 fingers, so not your pinky and not your thumb. Take those middle 3 fingers and touch your fingertips simultaneously down onto the 3 pennies, like 1 penny per fingertip, okay? And the question is, when you do that, what do you feel? What do you experience? And most people, they actually feel all 3 pennies as cold. Even the middle one, your middle finger, gets a cooling sensation. It feels cold. And here's the crazy part that makes it even more fun. You can now like spread your fingers wide apart while still touching the pennies, like just slide them on the counter a little bit apart while still under your fingertips. And what you'll find is the middle penny will still likely feel cold, even when it's more than an inch away from the actual cold pennies. Our brain is great at localizing an acuity for pressure and even kind of for vibration types of touch information sometimes, but it really sucks at acuity for temperature. Since the fingers are normally pretty close together in space, if your index finger and your ring fingertips are both cold, your brain is going to expect your middle fingertip to also be cold. So that's what you may perceive. Even though it's not touching anything cold, it'll feel cold. You'll perceive it. Your brain will construct that experience because of those expectations and what's going on in the two nearby fingers, which your brain knows are right nearby your middle finger usually. So we aren't built to have that much spatial precision for something like temperature. There's actually a lot of convergence, and the receptive fields for what our brain neurons distinguish for receptors, for thermoreceptors, are often pretty big. Okay, now before we move on to the last topic of this longer video, I wanted to get a little philosophical for a moment and show you some cool technology. So this little 3-minute video clip that we're going to watch, which is now pretty old, the tech's been around for more than 10 years, it's for a device called the Brain Port. It's a sensory substitution device. Basically, for someone who is blind, one creative solution is to have a camera take in visual information, and then a computer translates that into some sensory system that the person does have intact. Kind of analogous to cochlear implants, except with cochlear implants, we're just using technology to stimulate the auditory system directly when those sensory receptors aren't working properly. But here, instead, you'll see that we're bypassing the original sensory system entirely, and instead just translating the information that would have gone to that sense into another sense that the person still has. So that's sensory substitution. See what you think as you watch this video.
Your brain is what really sees, not your eyes. If your eyes don't work, and you can create another portal into the brain, then your brain is what is going to interpret the world around you. BrainPort is essentially a camera that translates a video image to a plate that I wear in my mouth. Dad. Hundreds of pixels on the plate tingle on my tongue, and together they form patterns and shapes that my brain interprets as the space around me.
The information is the same whether you collect it from your eyes or from our cameras with a tongue display. He knows a lot about his environment already through the sense of sound and touch, and this adds one more piece to the puzzle.
I think it's amazing that I can look on the screen and see that his brain is tracing a number and able to figure out what it is.
31.
Very good.
Stone, paper, scissors, shoot. I'm just at the beginning of this stage, but I think you could take this innovation so much farther than probably anyone knows.
Now you can zoom into what you just did and find your O.
I'm laughing because I think I beat you.
Just to be clear for a moment here, he's not seeing what's on that little screen. That little screen is for the purpose of the researchers or his family members to see kind of roughly how the camera is picking up what he's looking at. He doesn't actually get that sight that's on the screen. Rather, it's just an array of electrodes on that little lollipop on his tongue, where if it's, let's say, an X that he's looking at, an X that the computer has picked up, it just means the electrodes on his tongue will kind of go off in an X pattern. So it's really just a touch on his tongue in an X shape. It's an X-shaped touch on his tongue that his brain learns to interpret as, then there's an X shape out there in the world that the camera's looking at.
I thought I had a circle on the left, circle in the middle, and a circle. down at the bottom. But didn't I have a circle on the top left?
No.
You stinker, Emma.
Oh yeah. We both won.
No, we didn't win both win. I can see it. I can see it, I just can't grab it. It's just a trip for me to be able to look at a hold and reach for it. It's a trip for me to be able to have Emma put her hand out and me be able to see what she's doing. To me, it's like beyond science. It's wild. It's a wild experience.
So that's the brain port device that I was talking about again. Now, it's kind of mind-blowing, right? The original systems were actually developed to stimulate a person's back, kind of like pushing a simple image of the scene up against their back with little pens. But it has crappy resolution. Why? Well, we should kind of know that. It's due to the back's poor acuity and larger receptive fields for touch there. So this version, this lollipop kind of version of the brain port, this works much better since it uses the tongue, a body part with very fine-grained acuity with small receptive fields and lots of S1 neurons dedicated to it. And in the S&P literature, sensation and perception psychologists and other neuroscientists still actually debate and do experiments to try and determine if we should consider this sensory substitution to be giving the user actual vision. Like, is it visual perception via just touch input? Or whether it's more akin to a touch experience, but just allowing touch at a distance? Or whether it's something kind of unique and between them or distinct from the individual senses and perceptual systems that are involved. Give it some thought and see what you think. Another example, kind of along these lines, is the feel space belt, which it vibrates in the direction north is. So you put this belt around you, and as you're moving around, turning left and right, and imagine, you might be a person who's blind, or it might just be you're out in the woods and kind of things all start to look the same. But you always have this vibration of which direction north is. So it basically has a bunch of little motors on it that make a cell phone-like vibration that tracks north as you move, as you turn around. Once people get used to it, some users start to describe it as feeling, that is perceiving, compass direction, which is in fact something that some other animals do. It's called magnetoreception. It's a sense that humans don't naturally have. Some other animals do. But it's still coming in through touch receptors and the touch pathway. So is it truly a sense of direction? Do people wearing this gain a new sense, a sense of direction or a sense of magnetic north? Or is it honestly just our normal, usual sense of touch, and we're using the touch as an information channel the way an audio app on our phone could tell us with words or sounds whether we're facing north? Again, maybe just give it some thought and see what you think. Now, meanwhile, I want us to hit on one last topic for touch perception before we end the somatosensation topic. What we've been talking about so far for touch perception is focused on how accurate our touch system is for things that push up against our skin or vibrate our skin or whatever. And this is what we might call passive touch. It happens without you having to move a muscle. You can just feel if something touches you. But a lot of our touch perception is not just passive touch, but what we might call active touch, where we deliberately move our hands or other parts of our body against a surface or object, and in doing so, we gather a lot of sensory information and have a touch experience. So let's talk about active touch a little bit. If passive touch is basically objects or surfaces just coming into contact with us, then active touch, which is often called haptics, the field studying it is called haptics, is when we explore or identify objects using active movement. We manipulate things in our hand, or run our fingers along things, or grasp or heft objects, and use that info to figure out what it is and how it works, what its affordances are. And that's a very active process. It requires sending out motor signals to make movements that'll gather new information for us. kind of analogous to a chess player who's looking at a chessboard but decides to stand up and move around to circle the board from a different angle. Movement allows collection of new sensory input in an active process. Now, we use active touch all the time without even thinking about it, including when exploring a new object in our hands, or especially when feeling things you can't see, either because it's dark or they're out of sight, like around a corner, behind the couch, or whatever. And we're pretty good at it. In fact, in laboratory tests, it's been found that we can identify most objects haptically, using touch, in about 1 to 2 seconds. Very fast. Actually, Susan Lederman and Roberta Klatsky are the absolute pioneering experts in this field. They've published tons of papers on this, and back in a landmark paper in the 80s, they identified a set of human universals in terms of how we do haptic identification. Actually, these are pretty similar to how our primate cousins do it. So a friend of mine in graduate school did a study on monkeys and how they explore objects in their hands when they can't see them, and it's very similar. But these are kind of human universals in how we use haptic movement, gather haptic information, moving things around in our hands. The actions we take when trying to identify an object using haptics alone, they're called exploratory procedures. And there's basically a limited set of these procedures that we all use quickly and automatically to make sense of things in our hands. If you watch kind of video of people exploring things in their hands with their eyes closed, you'll find we all do very similar things, especially to identify particular things about the object. So the exploratory procedures we use actually depend on our goals when exploring the object. Certain exploratory procedures give us texture info, like the contour following and the lateral motion that you see here. Lateral motion meaning like rub your fingers along the surface, which we know would activate Pacinian corpuscles through vibration, and we know that's important for texture perception, right? So it makes sense why lateral motion would be one of the exploratory procedures to gather texture information about an object. Meanwhile, other exploratory procedures like enclosure of the object in your hands combined with contour following, that gives more general shape information, and so on. So there are others. I'm just showing 4 of the exploratory procedures here. There's a bunch of others, but different exploratory procedures in different combination will give our brain all sorts of new info that we wouldn't get if someone just set the object onto our open palm up hand, where all we would have is passive touch information. And we know the brain uses this information because a lot of neuroscience work has gone into identifying how the brain uses input from those exploratory procedures. For example, some neurons later in processing, so further outside of S1, when we get into those supplementary areas nearby, we find some neurons that don't just fire for touch to one specific Merkle disc or something like that, but for more interesting combinations of those simpler firings, combinations of those earlier neurons in S1 going off. So for example, there will be some neurons later in processing in other parts of the brain that'll go off specifically for feeling edges of a particular orientation, a particular angle in, say, one specific part of your hand. In other words, like this monkey hand we see here from a study that, again, used an animal model, there's a neuron somewhere in its brain out past S1, so maybe an S2 or a nearby area. There's a neuron that'll fire only if there's a horizontal edge at an angle, like the angle we see here with the red line. If an edge is close to that angle, like it's a little bit off, right? The angle is a little bit tilted, like a big diagonal. This neuron might fire a little bit, not as much, right? But it fires maximally for an edge of just that orientation, kind of going side to side along that part of the hand, which obviously will happen when doing a given exploratory procedure on some objects, but not others, like holding a ruler sideways or something like that. And again, there's one neuron somewhere in the brain that'll go off for that kind of specific situation. A different neuron might be honed to go off maximally for this orientation. A different neuron might be honed maximally to go off for that vertical orientation, an edge that goes up and down that you feel across your hand there. There might be another neuron watching for a horizontal object, a horizontal edge down at this bottom part of your palm there. Another neuron or something that might go off only if you get kind of a straight edge across both of these creases here near the tips of your fingers, a variety of neurons later in your brain that will look for various combinations of things, but that might fit these more realistic inputs that we'll get when we do exploratory procedures on entire objects, not just a tiny little Merkle disc getting stimulated, but a whole bunch of Merkle discs all in a row. We've got a neuron later in the brain that looks for that combo of all those Merkle discs getting turned on rather than all these other ones. Likewise, other neurons exist, again, outside of S1, further into the brain, but other neurons exist which have receptive fields for movement in a specific direction, like when rubbing your finger one way on an object or the object rubbing in that direction, but not much firing for the opposite direction, say. The combination of specialized neurons like this can help distinguish the info we get when we do these exploratory procedures in our hands. It helps the brain build up a representation of the object or recognize it from past experience based on touch alone without having to see the object. And there are other neurons, again, later in the brain, that only fire for particular combinations of those earlier neurons firing. So eventually we actually have neurons that only fire when grasping a specific type of object, like this ruler here, but not a cylinder, even with a similar grip in the hand. So for example, this one neuron, this example neuron in the brain of a monkey, fires when grasping a straight-edged object, the ruler, but not for other objects. The green graph here, the green lines graphed out here, that's the firing, that's the action potential, so the firing rate of that one neuron that's deep in the monkey's brain, and it goes off when the monkey starts holding the ruler, but doesn't go off when holding something else. So this neuron only fires for straight-edged objects, meaning that neuron can tell the brain, I'm holding a straight-edged object right now. And in combination with other neurons to help identify, then the brain will be able to say, oh yes, this combo of features, this combo of later neurons going off only happens for a ruler, a stick, a handle, whatever it is, different things. We can start to identify those objects. If you want to directly experience the difference between passive and active touch, there's actually a pretty easy way to demo it, again, at home with like a friend or a partner, if you've got a second person to help you out. So for this, you'll need some of those little foam letters that are often sold in a kid's aisle, like as a toy or a bathtub toy, or they may be in the arts and crafts section, but usually just for a couple dollars. Get a bunch of these foam letters. I mean, you don't need a ton, but just a variety of these foam letters. Ideally the smallish ones that are like, an inch and a half or so in height, and then mix them up real good. Whoever is being tested, you can take turns, but whoever is being tested, keep your eyes closed the whole time, and what you'll do is put out one of your hands, palm up and open. So you'll just be hanging your hand up there, palm up, waiting for something to show up in your palm, but with your eyes closed. And, basically just trying to make a big flat surface onto your palm that the other person can then press one of those foam letters into. And for the procedure then, the testing, that friend who's acting as an experimenter, they'll pick a letter or a number at random and just press it against your palm for a couple of seconds, then remove it. And you have to try and guess what letter or number it was. Tally up how many letters you get right out of as many tries as you want to do. And the experimenter should not be giving feedback until the end. Just don't give them hints. Don't tell them if they got it right or wrong until the end. But then the essential part here is repeat that whole procedure. So keep your eyes closed. Repeat that whole procedure for that many trials. Again, using a random letter or number each time. But now the experimenter just sets it in your hand, and you're allowed to move it around within your hand. So just in that one hand, move it around within, touch it with your fingers, rotate it, feel it, in order to identify the letter or number. In other words, you can use active touch in this second condition. And when you compare your performance, passive touch versus active touch, when did you do better? You'll likely find that active touch is insanely easy and way, way, way more accurate than passive touch. By the way, haptics is actually a really big field where psychology overlaps with a lot of other fields like engineering and medicine and computer science, virtual reality. They have lots of practical applications. So just as one example, haptic feedback is integrated into a lot of robot-assisted surgery machines these days, so that the surgeon who's controlling the robot doing the surgery inside of you, the surgeon gets feedback from active touch. It'd be kind of like if the mouse on your computer felt in your hand like whatever object you hovered over in a video game. So indeed, virtual reality researchers and commercial companies for VR have been working on haptic integration for decades now, usually just in a laboratory, in a research laboratory. But now they're starting to look at ways to open that up to get more haptic feedback, more complex haptic feedback into the systems we might use for video games and stuff like that. And just because we talked about hearing damage when we went into the audition topic, I think it's worth mentioning here that there are some conditions affecting touch perception, damage to our touch perception. And the most well-known of these is called tactile agnosia. There's an area of the brain, it's actually an S2, the secondary somatosensory cortex, up there in the parietal lobe, just behind S1. And in this area, this part of S2, if a part of it is damaged, there are neuropsychology patients, brain damage patients, who have lost the ability to recognize objects haptically. Like they can move it around in their hands and they can't figure out what it is. You can show it to them visually and they have no problem. They can tell you what it is. Even if they hear the sounds of it, they may be able to identify it. But put it into their hands with their eyes closed and they are at a loss. And of course, this can happen on just one side because touch info is processed contralaterally, right? So someone with right hemisphere damage to that S2 area might show tactile agnosia with their left hand because the right hemisphere controls touches from the left hand, or controls the left hand, gets touches from the left hand. Well, their right hemisphere performance, their right hand performance, if the right hemisphere is damaged, their right hand performance might still be pretty good. Their left hand performance, right, opposite, contralateral to where the brain damage took place, might be pretty bad. So just FYI from this term, agnosia, we're going to run into it again. When we talk about vision in some of our future topics, we'll run into that term agnosia a lot. So I just want to mention here before we move on, the root word here is gnosis with a G, gnosis, which means knowledge. And then the A prefix in front of it just means lacking something, not something. So you're probably most familiar with someone being agnostic. Like if they don't claim to have knowledge of a god or gods, they're agnostic. It's just an A, lacking, plus gnosis, knowledge, so lacking knowledge. So we use that term in neuropsychology for conditions where someone can't recognize or identify objects, where they can't demonstrate knowledge of things through that particular sensory channel. So here we've got tactile agnosia. We'll see later that there are visual agnosias, there's auditory agnosia, there's other types we'll talk about. Okay, finally, one last demo you can try with a friend. I'm going to call this one the dead hand illusion. You may have done this as a kid at like a sleepover or summer camp or something, because it's just kind of like a fun, weird experience. So here we're going to confuse the brain by giving some mixed signals to active as well as passive touch at the same time, simultaneously. So what you'll do is raise your hand up, like maybe raise your right hand like you're going to high five your friend, and align your right hand to their left hand, or vice versa. But basically like you're high-fiving where the hands are actually aligned, thumb to thumb, pinky to pinky, okay? So one of you might have to use your non-dominant hand or whatever. So it's like a stuck-together high five. You're going to keep the hands up there in the air against each other. Just hold the high five for a minute. Hold the hands against each other for a minute. What you want to do now, you can take turns for this so each of you can experience it, but one of you will use your thumb and your forefinger kind of spread out like you're going to squeeze something. Take your thumb and your forefinger and feel along both of the middle fingers, for example, or just rub up and down along both of the ring fingers. And again, you'll be rubbing the back of your own finger and the back of the other person's ring finger, or the back of your pinky and back of the other person's pinky at the same time, the back of that person's index finger and the back of your index finger at the same time. It's almost like you're going to like squeeze those fingers together between your thumb and forefinger, but don't actually squeeze. Just lightly feel up and down whatever merged finger pairing that you want and try it on a few different fingers. And often when you do this, people report that it feels numb or dead. And that's because the brain is not getting half of the passive touch input that it expects from the active touch hand. The active touch input of your moving hand, right? Because that's telling the thumb and the forefinger on your moving hand, the active touch stuff, that's feeling something as they rub on both sides, right? Your thumb and your forefinger, you're feeling both. You're feeling the other person's finger, back of a finger, and your back of a finger. So your brain knows that's happening in the same part of space where your own hand currently is. So you feel like you should be feeling that touch on the front and back of whatever of your own fingers is being rubbed. Yet one side isn't feeling that passive touch on the high five hand, but that's because it's blocked by your friend's hand. So your held up high five hand is only getting half of what your brain expects the input for it, that your other hemisphere is sure it should be feeling right now. Hence, you get the weird perception, which some people describe as like a dead hand feeling. It's pretty interesting. All right. That's actually going to finish our main topic for somatosensation, where we spent some extra time on touch, since that's one of the traditional five senses and pretty well studied. But remember, somatosensation also includes things like pain. And we didn't really talk about pain that much, except to see that those nociceptors are the sensory receptors for pain information. So actually, for the next topic in the course, we're going to spend a little more time on pain perception. And we'll hit that in our next video.